Hi guys.
Hey, Miss Claire, what's up? Oh, she was there. Now she's gone. There she is. Hey. You doing good?
Yeah. I wish I could be there. I want to dissect.
Ah, I'll see if I can get video of it. I should be able to. See if I can get any more volume. Sometimes I can get it to come through here, and sometimes I cannot. I don't know. Claire wants to be here because she wants to dissect things.
Are we dissecting something today?
We are, but it's a very small something. So it's no, it's not a lizard. It's a planarian, a flatworm.
They're literally like,
They're about this big.
Savannah caught a turtle.
She did. What kind?
You have no clue, but turtle? A turtle. We think it was the eastern like
Box turtle.
box turtle. Check the picture. And then they also caught a lizard.
Oh,
too.
we have a lot of those. I have at least 4 kinds of lizards in my yard alone that I know. What kind of turtle is it?
There's a lizard.
Yeah, that's a box turtle. That's an eastern fence lizard. Actually, an eastern box turtle and an eastern fence lizard. So the box turtles, so actually tortoises, right? Because they're not in the water. Like technically your testudines are divided into turtles, which are aquatic, and then tortoises.
It's gonna drown.
Yeah, like it'll swim, but it's not a swimmer. You know what I mean?
That's why we put 2 rocks in there.
Mm-hmm.
And like we put it on a rock and then they found a lizard and they put the obviously the lizard was trying to get
That's
out.
amazing. She caught that because those lizards are fast. Unlike the turtles, the lizards are super fast.
Yeah.
Yeah, well, the cool thing about box turtles is they're on the bottom of their shell is hinged. So like right about where their armpits is, there's a line that runs this way. And they're different from other tortoises in that they can take that front part of their shell and fold it up. So it's actually hinged like a little trap door.
When we were releasing him, I was the one who was holding him because we were
Mhm.
also saving frogs, so we would
Uh-huh.
step on them. 'Cause
Yeah.
When we released it, it was like 8,
Oh.
and it was already dark,
Yeah.
and we were like looking around for frogs, and I released it, and it was like the dip. And I was like, that's so weird.
Yeah, this part. Yeah.
Yeah.
Now the eastern box turtles, the tortoises are really easy to sex too, because their eye color, I think the males have, one is brown and one is orange. I think the males are orange and the females are brown.
Yeah.
And the other way to do it is on the bottom of the male's shell. Okay, so you've got the hinge in the front, the line here, and then down towards their abdomen, towards their tail, they actually have this like concave shape like this. It's like it goes up into them.
It had orange eyes.
Okay, yeah, but that on the bottom, it's actually because when they mate and they get on top of the female turtle, and it like fits like a puzzle piece, like her shell and then his shell has that little dip in it, and it's wild. And I actually caught 2 of them in my backyard. I've never even, like, you only ever see one turtle at a time. I actually caught turtles mating a couple weeks ago, and he was biting her head. Like he was leaning over, stretching his neck way out. His neck was like this long. It looked weird coming out of the shell, and he was biting at her face. Yeah. All right, let me make sure this is, yep, we're recording. All right, so we're gonna go ahead and start. Here comes Sky, and I expect the Armstrong kids to show up soon. So I'm gonna minimize this. Oops, I'm sorry, I didn't need to minimize that. So we do have 2 things. We have a new guy who should show up today. He's going to come a little later. His name is Anthony. I think he'll be here in time for lab, and he's going to try out our class, and he may just be joining us, which is pretty cool. Come on and slide up there, little thing. Okay, be like that and don't. And then we also have news. The Sabins, Annabella and Dylan, have gone back to public school. I'm assuming it's public. She said middle school and elementary school, so I am assuming that's public school. But anyway, they have left and will not be back. And I cannot make this do a thing here. Come on, do a thing. All right, trying to make this smaller. It does not want to go smaller for me. It's making me crazy. And it's in my way. Come on. Come on, look at that. It won't do it. Sorry. Zoom thing is like being picky. Will it let me drag it if I use my finger? What are you doing? Oh my gosh. All right, hold on. Switch it to me. Nope. Full screen. Again. Yeah, that's what I want. There, now it did it. All right, let me share my screen though so you guys can see what's up. What's up? All right, so we're starting cell division today, which is why our lab is such a good one. Our lab today, we're going to be dealing with planarians, which are flatworms, and they actually possess a special kind of cell, which is a stem cell, which is totipotent, which means it can turn into any kind of other cell. It's a cell that can differentiate. And we all start out as cells that differentiate because we all start out as one cell, and then eventually that one cell becomes all these different types of cells. So cells that can do that are called totipotent. And it's going to take 3 weeks for our lab to run. It's going to take some time to watch them regenerate. But because they have stem cells, they have the ability to regenerate entire body parts, kind of like a lizard that drops its tail or a starfish that loses an arm. We can't do that. Like if we cut our finger, we can, you know, grow a little bit of finger back like the skin, but if you cut your whole finger off, you're not growing it back, right? That's not going to happen. But these little guys can do it. What'd you say? I'm having a hard time hearing you today.
I saw a video about the flatworms.
Yeah, I did. I shared a video about the flatworms in case there was anybody who was like not going to be comfortable dealing with them. So we are going to talk about cell growth, division, and reproduction today. So there are limits to cell size. Now, why are there limits to cell size? There's some really good reasons for this. So the larger a cell grows, the more demands its regular running processes are going to have, right? And you remember when we did our cell membrane, things were crossing it, right? So the bigger and bigger it gets, the more things would have to cross that membrane, and the bigger that surface area would get, and it just becomes an awful lot for the machine to keep running. So when the surface area to volume ratio shrinks, so you got to think it's like a balloon with air in it, right? Sooner or later, you have more volume in the balloon than you have latex making the surface area on the outside. Same thing would happen with a cell. You would have more volume inside than you have membrane. And when that surface area to volume ratio shrinks, the membrane can't adequately accommodate all of the waste removal, and that's also bringing in nutrients. So what cells do when they get too big and they get overloaded with information materials is they divide. So we have what's called the cell cycle. And this is just sort of a graphic of the whole thing in one, right? This is the whole cell cycle in one. And it has phases. And you guys, for your homework, that's what it was going to be come up with some mnemonics for the phases. And some of them were already in there last night. I added them to the slideshow because they were fun. So a mnemonic would be like when you do King Henry died unusually drinking chocolate milk, right? And we know what those letters stand for. So they're, yeah.
I didn't memorize them.
Yeah. So the idea for the homework was to come up with a mnemonic for these different phases so you can remember what order they go in, and everybody could come up with their own, and some of them are really interesting. So process of cell division. So we have prokaryotic cell division. Now you remember what's different between a prokaryotic cell and a eukaryotic cell? What are you? Do you have eukaryotic or prokaryotic cells? Yeah, eukaryotic. And that means what? Do you remember what it means? It means that our nucleus is in a membrane, right? We have a concentrated nucleus inside of a nuclear membrane, and all of our genetic information is inside the membrane. Prokaryotic cells are really ancient, and they've got their DNA just kind of floating around in the cytoplasm, which is not how we do it, right? They're less organized. So the usual method for prokaryote cell division is termed as, we call it binary fission. And so break those two words down. What's fission mean? Do you remember fission? Fission. How about fusion? Fusion is the opposite of fission. Does that help?
I'm guessing it means take apart.
Take apart, because fusion means to put together, right? So fission means take apart, fusion means put together. All right, and then binary would mean there's two options, right? Binary code that we use in computers is 1 0 1 0 1 0 1 0 or on off, right? And do you remember how your daddy was doing it the other night? He was counting in binary. Like how can you count in binary?
Take up to 6.
You can count up to 6, but he did it like, do you remember how he did it? It was, wait, put your hand out so Claire,
Actually, no, I
Claire
did
can,
it.
make sure Claire can see it.
Exactly what he did.
Yeah, because it was kind of funny, so don't be offended. It's a real
I
thing.
didn't do it right, but he did. It was
1, 2, 3, 3, right? 4.
Was it? No, it didn't do that.
No, he did 1. How'd he do it? Oh shoot, I don't remember either. I'd have to have him come. He's the binary guy because he works in telecommunications. But he was showing how you could count in binary. And the reason we use base 10 for everything is because we have 10 digits typically, right? So, but binary works for computers. It also works for cells, which basically just means that the prokaryotic chromosome is a single DNA molecule. It's going to replicate. It's going to attach a copy to different parts of the membrane, and then the membrane is just going to pinch off. And that's what's going on in this picture here. We've got cells that are dividing. So each of those is a cell in the process of dividing. So this membrane would pinch right off, and then you'd end up with 2 cells where you had 1. Now, eukaryotic cell division, what we do is a little more complex. Well, it's a lot more complex. So it occurs in stages. So interphase exists in both mitosis and meiosis. And then we have what we're going to call mitotic phase. All right, these are like the umbrellas. Interphase is like your regular resting phase for a cell. So if you're a cell and you're not currently dividing, you're not currently doing something, you're in interphase. You're just chilling. It's just resting. And then within the mitosis process, mitosis is cell division, eukaryotic cell division. You have lots of steps, okay? Now, there are 2 words here you're going to hear a lot in the next couple of weeks, mitosis and meiosis. And they're very similar. One just has like an E and the other has a T, right? Mitosis is the one where you have cells just dividing to multiply. Meiosis is the process where they divide for the purpose of creating sex cells or gametes. So the difference is that in mitosis, you have a cell that is exactly the same. You have 1 cell, we're going to call it a parent cell, and it's going to create 2 daughter cells. And in that process, those 2 cells are going to be like clones. They're identical to the parent cell. But in meiosis, they're going to shuffle some DNA and some chromosomes are going to be divided up, and it gets really, really complex. And then you're going to end up with 4 cells that are all different from the one that started out, and they have half the number of chromosomes from the cell they started with. All right, so within your mitotic phase, you've got several different phases: prophase, metaphase, anaphase, telophase. And telophase with cytokinesis is kind of implied. Cytokinesis, that's a great word. So cytokinesis means the dividing of the cytoplasm. So it's when the cell's actually going to pull apart and literally cleave itself into 2. So for interphase, whoops, hang on, I didn't mean to click that yet. So for interphase, you've got the cell and it's just kind of sitting there, it's growing and it gets bigger and bigger and bigger. So I'm super proud of my animations. I have to tell you that I once drew something embarrassing teaching this lesson, so I went with animations instead. So interphase appears to the eye to be a resting stage, but it's actually a lot going on. I mean, of course, the cell's doing things. It's always doing things. They're indispensable activities and they make the next phase possible. So interphase generally lasts 12 to 24 hours in mammalian tissue. And during this period, the cell is constantly synthesizing RNA and producing protein. So think about that for a minute. The cells, like on your skin, for example, the ones you can see, well, you can't see the cells, but you can see your skin. They're only staying like they are for a half a day to a day. And then they're dividing instantly, poof, like that. That's kind of crazy to think that's going on all the time, right? So in mitosis, the next phase is going to be prophase. All right, so during prophase, the complex of DNA and proteins contained in the nucleus, which we call chromatin at this point, it all condenses. And then the chromatin coils around and it becomes really, really compact and it forms chromosomes. So your DNA is just kind of like a big knot in your nucleus. But in this phase, it kind of starts winding itself up, almost like you're winding yarn. Okay, think about like winding up a ball of yarn, all right? And it winds itself around and it forms these little chromosomes, which look like little X's. See them? Each one's like a little X, all right? And each chromatid joins with its sister chromatid. And there is a center point called a centromere. So this is a chromatid and this is a chromatid, and it's got a centromere in the middle. And together we call the whole thing a chromosome. Now, what we're going to learn more about when we start into like next week's lesson.
Is that where it comes from, XX and XY?
There's some, well, you have 46 chromosomes, okay? And we're going to talk about this next week. Really, this is next week's lesson. But you have 46 chromosomes in every cell, okay?
I was going to say, I only have 46.
No, in
chromosomes.
every cell. But when you look at these like this, see these guys right here? So one of these chromatids came from the male parent and the other chromatid came from the female parent. But they're what you call homologous pairs. We're going to talk about it next week, but it's like a pair of shoes. They're not identical, but they go together as a set and they find each other and they match up and they make a chromosome.
Does an
just
X
like this.
chromosome and a Y chromosome look the same? Or
They
does
look
one like
different.
an X and one look like a Y.
They look different. Yeah. The Y, well, we'll talk about it more when we get to it, but the Y chromosome, which denotes male features, is actually kind of, this is going to sound bad, it's a weak chromosome. It lacks a lot of space for genes that aren't there. So it's an interesting little chromosome. Because as much as it controls a lot of things that without it wouldn't happen, like it's the presence of testosterone and everything that gets kicked up by the Y chromosome that makes an embryo turn male, it's also lacks complementary genes that would go on the X chromosome that would keep it from having expression of certain sex-linked traits like color blindness and baldness and things like that. So anyway, the sister chromatids are identical copies of DNA joined at a point called the centromere. And so in this, the main stuff that happens here are your condensation of your chromosomes. You got to think about winding them up, right? Packing them together. And then these centromeres appear, these little guys here, they appear. And the formation of this mitotic spindle, that's those white lines, right? And the beginning of nucleoli start to break down. So in the center of the nucleus of the cell, it starts to break apart. So the next thing that happens is metaphase. All right, now in metaphase, the process that separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. So what happens here, you kind of see it. So what happens here is you've got all of these guys lined up. All right, see how they're lined up perfectly? Like soldiers, all these chromosomes. Now, you remember I said one strand came from the male parent, the other strand came from the female parent. But what happens next is, in this case, they're all kind of all the same. But anyway, it gets confusing. Off on another subject. So they line up here like this with their little centromeres in the center. And these guys over here, these guys are called centrioles, and they only appear when the cell is dividing. They're always there, but you don't see them unless the cell is dividing. Kind of like, what is it, Schrodinger's cat. But anyway, they form like poles, just like poles, like planet Earth has poles and magnets. It's the same thing, guys, exactly the same. We got a magnetic north and south sort of thing going on here. And so you get this mitotic spindle and it's fully developed, and your centromeres are at opposite ends of the poles. Centrosomes, sorry, are at opposite ends of the poles. And then your chromosomes are lined up. We call this the metaphase plate where they line up. Now you can think of these little spindles like ropes. And what's going to happen next is anaphase, and it pulls, they start to pull apart, and those little chromosomes get split in half and yanked to opposite ends, just like that. So anaphase is the fourth phase of mitosis, and it separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. The separated chromosomes are then pulled by the spindle to opposite poles of the cell. So what you had, so when we were kind of on a meiosis talk there when I was saying they were the same, because they're not the father mother thing is more when you get into meiosis, which is next week. So what you have here are two identical cells. So these are the same, The genes on all of these are exactly the same, okay? And you get cohesion proteins, which is what we call these. They bind the sister chromatids together. Those are the things in the center. They break down so that they pull apart. And now you've got these non-kinetic core spindles, which lengthen and pull them apart like this. And then the next thing that happens is telophase. And telophase is when the membrane starts to come around and it's going to actually pinch off. So telophase, the cell undergoes a process called cytokinesis that divides the cytoplasm of the paternal cell, or sometimes they just call it the parent cell, paternal in this case, meaning just parent, not necessarily like father, into two daughter cells. So your chromosomes begin to decondense. See how they're starting to kind of float around in there now? And your spindle breaks down and your nuclear membranes and see, here's your nuclear membrane reforming. So it's just becoming just what it was again. And then you get cytokinesis and it breaks apart. This phase right here where it breaks apart, they call cleavage. And this is called a cleavage furrow. Cleavage as in cut, like with the meat cleaver, chop, split in half. So cytokinesis is the physical process of cell division, which divides the cytoplasm of a parent cell into two daughter cells. And there's a contractile ring. That's what this is. It's like going like this. Okay, it's a contractile ring and it shrinks at the equator of the cell, it pinches off the plasma membrane. So you think about like you squeezing a hose. It's the same kind of thing. It just squeezes till it's all the way closed. And that's how they divide. So here is, Claire, what's up? Hang on, let me pause this so I can hear you. Yep, go ahead.
Yep. How long does the cell cycle take? How long does it
take? Okay. Well, it varies, but I don't know what an average is. Let me see if there's an average. It's not very long. I don't know. Let's see. I think it would depend on what kind of cell. Let's just say somatic. Those are your regular body cells. Let's see. How long? I'm wondering. That's a great question. Because it happens so frequently. Oh, I'm having this internet thing where my phone doesn't want to like do anything. Don't know why. Come on. I think maybe our new friend is here. We have a new friend showing up today. Yes. Would you go introduce yourself real quick? Sure. And Claire, hopefully this will load up. Anthony, hi, come on in. How are you? This is Drew. Hi. Drew lives here. In 1 second, Claire, okay? Ozzy, hush. Anthony, you're welcome to just come in and have a seat. And Ozzy. Oh, he's Ozzy. Yeah, that's Ozzy. Did you really? He's annoying, but he'll be quiet in a minute. He just barks when people come in, and then he's chill. Yeah, sure. All right. So, Anthony, this is Claire right here. Wave, Claire, for a second. Can you wave? There you go. Okay, and then Sky is on as well today. We just have some people that are out today, so they're all online. Claire, I'm gonna find that answer and get back to you because it doesn't want to load up. Okay. Claire had a question. And then we have Darcy and Lewis who are also out right now. So this is your first year homeschooling, Anthony? Is it your first? Yes. Did you do last year online or with the? I did last year in person. Oh, you did? You went in person? You braved the COVID school. Was it good? Did it work? Meh? Kind of. Yeah, sort of. All right. Well, everybody here has kind of had a different track. Like Drew's been homeschooled since first grade, halfway through first grade. Lily, you were about the same, weren't you? Second grade. Were you all along? All along for Colson. Claire, all along. I don't know about Darcy and Lewis, and I know Sky got to like third, maybe.
I went to I was the only kid in the family to go to actual pre-K.
You went to actual pre-K. Okay, cool. I don't know why, but Claire's very quiet today. Can't seem to make my speakers louder. All right, cool. Excellent. So we were just about to watch this video on cell division. It's just a few minutes.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day, our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into three distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
So there we go. So Claire, they gave you an example of 10 to 20 hours. So I was just looking while you were there because I know like skin cells go faster, for example, like they said. So what I found was that I was hoping to give you an average, but there's really better to give you a range, somewhere between 1.5 and 20 hours, depending on the type of cell. Plant cells tend to be obviously slower in the 10 to 20 hour range for an entire mitosis process, but skin cells go very, very fast, which is how you know, thank goodness, because you heal. Plus you're always banging stuff. Do you know what dust is in your house? Do you know what it is? What is it?
Dead skin cells and dust mites.
Yeah, mostly it's your skin. Gross. It's mostly your dead skin cells that have fallen off. Ew. Okay, so here we go with our mnemonic devices. So your homework was to come up with a mnemonic device to remember.
We'll get that tomorrow.
Yep.
I promise you will.
I know you will. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis, right? So I asked you guys to come up with some sentences that could help you remember it. So this one was from Lewis, and I only pulled off the ones that were available last night. So if you hadn't done your homework before, like, I don't know, 6 o'clock last night, I didn't get it because I just hadn't gotten back to it yet this morning. So he had intelligent people make all things clear. That's good. IPMATC. Okay, some people remember PMAT, and they just always remember that the I's in the front and the C's at the back. Now, Darcy's is interesting. Darcy's is improper poopers must always think clean. And Drew responded back to that on the discussion with how does one poop improperly, which I thought was a good question. But improper poopers must always think clean. And Drew came up with, I punched my annoying tennis coach, but whatever works, right? Whatever helps you remember. These are all good. These are all good.
Everyone was gonna put something in similar to that.
I punched my annoying tennis coach. Well, you know, please excuse my dear aunt. Y'all know, please excuse my dear Aunt Sally for order of operations. Parentheses, exponents, multiplication, division, addition, subtraction. There's a caveat to it that people forget. And my 7th grade teacher nailed it into our heads. She always said the words last remarks. So it was, please excuse my dear Aunt Sally's last remarks. And the last remarks meant from left to right. Because when you look at it, you know, you have parentheses, exponents, and then multiplication, division. And it's not multiplication and then division. It's multiplication or division from left to right, whichever is first. And then it's addition or subtraction from left to right. But there are people who will stick to please excuse my dear Aunt Sally and assume that multiplication always comes before division. And it doesn't. It only does if it's left of it. So there's that fun little thing. And then also Colson's was, I play Minecraft at the computer. That's a good one too, actually. That's a really good one. I can remember that one. Awesome. So we're talking about cell division. And if we're talking about cell division, there's also an important thing to consider: what causes cancer? Okay, cancer is a problem with cell division. That's exactly what it is. So I have a little video on what causes cancer too.
5G.
From a normal
Not 5G.
cell to a cancer cell, take 5 minutes to get the picture. More than 60,000 billion cells make up the human body. They are the units that form the tissues which make up our organs, like the liver, heart, and lungs. When the body needs it, our cells split into two and replace those cells that are defective or have come to the end of their lifespan. This makes it possible for our tissues to preserve their shape and their respective functions with the passage of time. Every cell is therefore programmed to multiply and die. This ordered but complex program is controlled by the center of the cell, the nucleus, which contains chromosomes containing many genes made up of DNA. Sometimes some of these genes undergo a change. The nucleus then sends out abnormal orders and the cell goes wrong. It multiplies uncontrollably and takes on a life of its own. Each new cell produced contains the same defect. The cells proliferate chaotically and form a tumor. This process may be short, but is often long. 10 to 30 years may separate the birth of a first abnormal cell from the appearance of a tumor of about 1 cubic centimeter. The tumor forms many blood vessels in order to survive. These will supply the tumor with oxygen and nutriments, allowing it to live and grow. This is what we call the phenomenon of angiogenesis. The tumor, however, only really becomes dangerous when the cancerous cells begin to invade through the vessels into the adjacent areas and spread to the surrounding organs. These cells can then invade other parts of the body, multiply and produce new tumors. Metastasis is the term used for this spreading process. But why does a cell become cancerous? There are many factors, and they can exist to different degrees. Factors have been identified, such as hereditary genetic anomalies, exposure to some viruses, HIV, hepatitis B, C, and D, papillomavirus, Epstein-Barr virus, exposure to toxic agents, chemical products, radiation, sun, unhealthy behaviors, such as the consumption of alcohol and tobacco, or a diet too rich in fat and low in fruit and vegetables. Today, over 25 million people in the world live with cancer. 7 million die from it each year. The disease represents the leading cause of mortality for those under 65. Lung cancer is the most widespread, followed by breast cancer, colorectal, stomach, and prostate cancer. The means to combat cancer do, however, exist. These often complementary treatments are used alone or in combination according to the type of cancer and its status. The goal of the treatments is to make it possible to eliminate the tumor and cure the patient in early stage cancer, or like a chronic disease, to control its development. There are mainly 3 kinds of treatment. Surgery consists of removing the tumor in part or in its entirety. Radiotherapy exposes the tumor to radioactive rays, which prevent the diseased cells from multiplying and destroys them. Chemotherapy consists of administering systemic medicinal substances to destroy the cancerous cells or prevent them from spreading. Chemotherapy and radiotherapy act on cancerous cells, but also on healthy cells, which explains their side effects. But today, better knowledge of the characteristics of cancerous cells makes it possible to develop therapies that target the cancer development mechanisms more specifically. Sanofi Aventis R&D is already present in all these areas to fight against cancer on all fronts.
So cells are programmed to die. They are. And if they don't, bless you, sweetheart, they don't die when they're supposed to die, they will grow out of control, which is how you get cancer, how you get tumors. So you hear about cancer a lot, but you don't often think about what it is. That's exactly what it is. Cells that won't die when they're supposed to die. So we're going to take the last couple minutes. We're going to talk about some cell differentiation. And then, of course, we are about to wrap up Unit 3. So we will go over our Unit 3 topics for our Unit 3 test. So a zygote is a brand newly fertilized human or animal or plant or whatever. So basically, when a sperm and egg come together, what you get is a zygote, and it's one cell. And then it will begin to divide. And once it starts to divide, they call it an embryo. So it just is only a zygote for a tiny amount of time. And then it becomes a 2-stage embryo, a 4-stage embryo, an 8-stage embryo. And you can see, you go from 2 to 4 to 8, right? And then you get to this stage where there's a lot of them in there, 16 or more, and they call it a morula. And then what happens is really, really cool. So you get this blastocyst, which basically means the cells will take this shape. They'll leave a little bubble area here. They'll make a little line around the outside. They're still within this sort of a membrane, okay? And they will all gather together except for this little, looks like a basket handle. Like this one's upside down, but it doesn't have to be upside down. And they call it a hatched blastocyst when it comes out of that membrane. And then it'll implant in humans or mammals, it'll implant and begin to grow. And all of these cells will then they'll start to take a shape, and it'll go into this little ball shape. And then this super cool thing happens where it'll start to fold in on itself, kind of like this. It rolls in on itself. And when it rolls in on itself, it's actually differentiating types of tissues. And there are then layers. So if you get into studying embryology, it's really, really fascinating, because at this point, there are layers of cells within this teeny, tiny embryo. And each of those layers has now already differentiated to a degree because they started out all the same. And now that it's in layers, then it will go from those layers to specific tissues. And when it gets to that point where it's divided into layers, you can do this really cool thing called fate mapping. And it works really, really well. If you do college biology at a higher level, have you done it or have you seen it?
Is this gonna be for the test?
No, not specifically. No. I'll show you what's on the test. Like always, I'll show you. So fate mapping is easy to do with amphibians. So if you get into higher level college biology, you may get to do it. And it's great to do with amphibians because their embryos develop outside of a body, right? Like they develop in water. You can see tadpoles develop in water from eggs. And you can actually go in and put a type of dye, that's a bioadhesive dye, on one of these cells, like at a 4-cell stage. You can do it. I've done it. It's crazy. You can go in and you put a tiny bit of dye like on this cell right here. And then that dye will stay with that cytoplasm pretty much forever. So as it's a bioadhesive dye, the one we use was called Nile blue. So it's a nice blue color, like a royal blue. And whatever this cell divides into will be that color. So as the embryo develops into a tadpole and then a frog, part of that frog's body systems are going to be blue. So maybe the one you tagged differentiated into a nervous system, or maybe it became skin, or maybe it became digestive system. But you can watch it develop, and it's so cool. So I'm going to show you one other video, and this is a fast video, but this is human embryos under a microscope. And this one is really cool. This is actually human embryos being fertilized.
Human embryos,
it's cool.
Under a time-lapse microscope begins like this. A human egg, as shown here, is fertilized by either conventional IVF or intracytoplasmic sperm injection.
So this is IVF, which is when they do fertilization in a lab, or in this case, like, see, they're actually injecting a sperm into the egg. Look, see?
At 16 hours post-fertilization, the egg highlights signs of fertilization as the male and female DNA come together. At around 24 hours post-fertilization, the fertilized zygote undergoes the first division to form a 2-cell embryo. The embryo continues to divide, and 2 days following fertilization, the embryo reaches the 4-cell stage.
Now we're at about 2 days. 48 hours right there. Isn't that wild to see? That's really cool. I've seen frog embryos do that for real, but
And
not.
on day 3, it becomes
not
an
human.
AA stage embryo. The embryo grows further, dividing from 8 cells to 16 cells, and from 16 cells to 32 cells. The embryo then makes the transition to reach the motherless stage.
So there's your morula. It's like a big
The
old
motherless
clump.
stage embryo progresses.
So now you're gonna go
Further signs of
blastocyst.
dilatation are seen. Blastocyst formation takes place.
See how it makes that big hole inside? Like a bubble.
The embryo
Everything
is
on
at
one side.
blastocyst stage on day 5. At this stage, the process of implantation begins.
Very interesting to see. Like I said, I've gotten to see it with frogs, but not with mammalian embryos, but with frogs, it's still cool. It's really cool. So these are your, if you wanted to check your homework answers, here they are. And of course they'll be, you can always go back and look at this because it'll be in the folder for you guys. And I want to be sure that I get you guys to your test topics. So pencils ready? You're gonna write down your test topics. Obviously you don't have to take a test, Anthony. You've missed everything, but all right, or you can get your camera ready, whatever works. So your unit 3 test topics are going to be stimulus and response. Remember that living things react to stimulus with a response. Active and passive transport, that would be across the cell membrane. What's active versus passive? Remember, active requires energy, passive does not. A good example of passive would be osmosis or diffusion. Steps of cell division, which we just memorized with, what is it? I punched my annoying tennis coach. Improper poopers must think clean, all that good stuff. I play Minecraft at the computer. ATP and cellular energy. Remember that ATP is our source of energy. ATP comes from ADP. And remember, it only lasts for just a second. And then it's gone because we've used it. And it takes ATP to make ATP. Photosynthesis versus respiration. What's the big thing when it comes to those two processes? What are they? They are completely photosynthesis versus respiration. If you wrote them out chemically.
Photosynthesis, you get it from the sun, respiration, you get it from the air.
Chemically, what are they? If you looked at the two chemical reactions, they would look like opposite, the complete opposite, right? Complete opposite. Remember that. Okay, prokaryotes versus eukaryotes. We said that today. Remember, eukaryotes have nuclear membranes binding their chromosomes together, as opposed to prokaryotes, which are ancient and do not. Plant cells versus animal cells. Big differences. Plant cells have what? 2 big things. Animal cells don't, but you'll see. What are they? Well, 3 big things. What are they?
Cell walls.
Cell walls, Claire. Yep. How about big central vacuole, right? And also chloroplasts. You won't see that in animal cells. Organelles and their functions. Remember, we had a nice big slideshow about organelles and their functions. Go back and look at the slideshows from class. Those are usually pretty helpful. Organization, cells to organism. How does that go? So you've got atoms that are elements, right? They make up molecules and compounds. Those make up what? Those make up cells, well, proteins and whatever. And then you get cells. Cells make up, somebody help me. They make up tissues. We're doing like what makes up what. So cells make up tissues. Tissues make up, somebody knows. Organs, right? Organs make up organ systems, and systems make an individual organism, right? That's organization. All right, good deal. Taxonomic hierarchy. All right, remember this one. Remember our taxonomy? We did it a little bit. This is like where you talk about prokaryotes and eukaryotes, and then we have archaea and eukarya, and then eukarya breaks down into kingdom, phylum, class, order, family, genus, species. Remember that? We're going to be doing more of that too. And then binomial nomenclature, what's that? Remember what that is? That's when you name an organism by its genus and species. Like, remember we had, some of you guys did the this thing. We created our own. I think Lewis's is over here. Oh yeah, this is Darcy's. Remember? We came up with a genus and a species for this make-believe creature based on what we were seeing, how we described it. Classification, the idea of classification, grouping things together by like characteristics. All right, producers, decomposers, consumers, scavengers, heterotrophs, and autotrophs. What's an autotroph? You know, Lily, you know what an autotroph is. Autotroph makes its own food. An example would be, yeah, plants. Heterotroph has to eat something else. So, yep, anything that's not photosynthetic, basically, and not chemosynthetic. So DNA base pairs. A goes with, what's A's partner? A goes with, and in DNA, so you have 4 letters. You have a C, a G, a and a T. What does A go with?
Oh, T.
T, and G goes with C, and vice versa. Until you get into RNA, and then the T goes with the U. All right, Tinker Day real quick, just so you know. It is October. All right, so those of you who are new,
I love Tinker Day.
my college has a holiday every fall called Tinker Day. Nobody knows what it is. It's a secret every year. It's always different. It's some morning in October, and they cancel class and hike a mountain. We will not cancel class, but we will do the other thing they do, which is eat Krispy Kreme donuts. So whenever Tinker Day falls, I will have Krispy Kreme donuts that week. Like, they will be here and ready for you. So if Tinker Day falls on a Thursday, we will still get donuts on Tuesday, because that is what we do. And anybody who takes a picture of themselves wearing green and gold on Tinker Day and sends it to me will get extra credit. That's the way we roll. Lastly, second quarter lab teams, here they are, except for Dylan and Annabella, because they are not here. No, second quarter starts next week. And we'll put Anthony in one of the places where Dylan or Annabella left, because they decided to split on us. So that's done. And this is going to be, you can go back and look at this. I'm going to attach it to the email, but this is just keep yourselves organized, basically. All right, Miss Claire and Miss Sky, I'm going to let you guys go because
Miss Danny,
lab will
Miss
take 2
Danny?
weeks. So it'll take you some time. Everybody's going to hand me an individual one. I can't pull
Miss
you up. There you go.
Danny,
Yeah,
I don't get the weekly emails.
you should now. I update you. I had your mom's email in for you because she didn't give me yours until just now, but I fixed it last night.
Okay, thank you.
All right, catch y'all later.
Question, what is the test gonna look like?
It'll look like all the others. Like, you mean how many multiple choice, how many true false?
Is it like multiple choice?
It's all the above. I'll look for you in just a minute when I get everybody out of here. All right, we're gonna end this and we're gonna go over for lab. If you need this stapler, it's out. If you want to staple your lab, you are going to want to be sure that you return your lab to your binder because it will take 2 weeks to get through this lab.